So now we really get into the exciting stuff. We're addressing how the antigen-presenting cells present their antigens to the lymphocytes. So antigens, or immunogens, are substances that provoke an immune response. We often use the abbreviation AG for antigen. The property of behaving as an antigen is called antigenicity, and the words antigen and immunogen are basically interchangeable. To be perceived as an antigen or immunogen, a substance needs to meet certain requirements. It needs to have a certain foreignness, a certain shape, a certain size, as well as being accessible. Antigens are not normal constituents of the body. They're usually molecules of complex composition, like proteins. These complex molecules are more immunogenic than simple polymers. For example, you may recall from general biology that things like proteins are much more complex because each amino acid has a different shape to it, versus a large but repetitive molecule like a polysaccharide, having several of the same monomer subunits. The small molecules do not generally catch the attention of the immune surveillance system, whereas these large complex molecules do. Even large repetitive molecules have less antigenic effect. So we're really looking at these large, more complex molecules having antigenic effects. Things that are generally antigens are proteins and polypeptides. There are things like enzymes and cell surface markers, or hormones that we find in circulation, as well as some exotoxins. Lipoproteins can also be antigenic molecules. We found those a lot in the cell membranes. Glycoproteins are found as blood cell markers. Remember the ABO blood grouping systems? Those are glycoprotein markers. Also nucleoproteins, which is DNA complex to proteins. So not pure DNA, but DNA complex to proteins can be antigenic molecules. And polysaccharides and lipopolysaccharides. These are found in bacterial capsules and bacterial cell membranes, so they make great antigenic molecules. They're nice and complex and easily recognizable. So again, substances have to catch the attention of the surveillance system. Most are large, complex molecules. Being large is not enough, as we've covered. Proteins are large and complex because of the different nature of each of the monomers. Amino acids have a great variety of structure. Something like glycogen, though, is a very large polymer, but it has a highly repetitive structure, repeating units of glucose. Pretty simple overall. So these are not usually antigenic molecules. Now, lymphocytes recognize and respond to only one portion of these really large molecules. This is the epitope region. So the epitope is the primary signal that the molecule is foreign. We'll watch a short animation here in a moment to show you the effect of epitopes. Sometimes we'll see very complex or mosaic antigens, which have numerous component parts, each which elicit a separate lymphocyte response. Again, this will be covered in the animation, so it'll give you a great visual of how that works. Now, another type of antigenic molecule can be very small foreign molecules, but they can't do the job on their own. They consist of only a very small determinant group, but because it's so small, it sneaks by the surveillance system. So it's only when the haptens are linked to a larger carrier molecule that there's some immunogenicity. You can see that in this figure below. Here are some small molecules all on their own. There's no antibody made. However, if we bound the hapten to a carrier molecule, this blue molecule here, then we see that an antibody is formed in response to this hapten, or very small molecule. Haptens are things like drugs or metals or household, industrial, and environmental chemicals. And they will bind to things like serum proteins. We'll explore this interaction a little bit further in the next chapter. But before we move on, let's take a look at a quick animation of these antigenic determinants, or epitopes, on antigen molecules.
Antigens are macromolecules, usually of molecular weight greater than 10,000, such as proteins and polysaccharides. They are recognized by the immune system as foreign. Individual antibodies are not made against the entire antigen molecule, but rather to particular chemical groups on the molecules known as antigenic determinants or epitopes. Many different antibodies can be made against a single antigen, each antibody reacting with a different epitope. Complex structures, such as the surfaces of bacterial cells, may have many different epitopes. Each different antibody binds only to the correct epitope.
So let's have a quick concept check before we move on. What term refers to a small molecule that must bind to a carrier in order to become immunogenic? Is it a superantigen, an isotype, an epitope, or a hapten? So a superantigen we can cross off the list. It actually does have some significance here, as superantigens are the type of antigen that develop toxic shock syndrome. We may have time to look at that later. An isotype, an epitope, or a hapten. I'm going to go between these two, epitope and hapten. An epitope was a region of a molecule, as we just saw, and so we can cross that one off the list too. And hapten then would be this small molecule that must bind to a carrier in order to become immunogenic. So there's a lot of cooperation in immunity throughout the system. We learned about a number of different immune system organs, as well as our lymphatic system. So the basic immune response involves an encounter between a leukocyte and an antigen. As we're going to see, it's a little bit more complex and there are multiple stages. But microbes and other foreign substances are going to enter mostly through the respiratory or the GI mucosa. They have other points of entry, but they're much less frequent. If these antigens make it into circulation or intravenously, they're trapped by the liver and spleen or the bone marrow, the kidney, or the lung. Other antigens are going to be carried in lymphatic fluid and concentrated in the lymph nodes. Now, the lymph nodes and spleen are really important in circulating antigens thoroughly. It's these organs that are responsible for exposing these antigens to the maximal number of lymphocytes in order to make sure they reach up with the connect clone to elicit an immune response. So as we'll see, first, the antigens are picked up by antigen-presenting cells, and then those antigen-presenting cells will circulate through various reservoirs of lymphocytes, and hopefully they'll run into the right clone so an immune response is elicited. So how does this antigen-presenting system work? As you're already familiar, the antigen is going to somehow make its way into one of these lymphoid organs, at which point it might run into an antigen-presenting cell. These are things like macrophages that go around gobbling things up, B cells that gobble things up, and then dendritic cells. So those are our three types of antigen-presenting cells. Once contacted, the antigen has to be formally presented to the lymphocytes. So the antigen-presenting cells pick them up and present them to a variety of lymphocyte clones until they run into the right one to elicit a response. But here we have one of our antigen-presenting cells. This one's a dendritic cell because it's often dendritic cells that pick up the antigens. Here is a foreign microbe. The dendritic cell will engulf the foreign microbe, and that foreign microbe will then be broken down in a lysosome into its constituent parts. The dendritic cell will then excerpt this antigen region or an epitope region from proteins within that microbe and present it on an MHC2 receptor. So as you can see in this insert, we now have an MHC2 receptor here in brown, and it's holding on to a piece of a microbial protein, the particular epitope that it's interested in presenting. So essentially, the role of the antigen-presenting cells is to engulf the whole microbe, or the antigen, and modify it so it's more immunogenic and thus recognizable to the T lymphocytes. After processing, the antigen is bound to that MHC on the outside of the dendritic cell, so it's accessible and recognizable by T lymphocytes. And then this is where it gets exciting, or maybe a little bit confusing. It could take a moment for you to sort this stuff out, but spend some time with it and I promise you it'll all become clear. But the antigen-presenting cells, in this case, are going to activate a T helper cell. Remember, they have different surface markers on them, and one type was the CD4 T helper cells. These are found in the lymph nodes. This class of T cell has an antigen-specific T cell receptor. You'll recall that these T cells are developing in the thymus, where they're going through a clonal selection process to get any one of a number of billions of different possible receptors on their surface. So now we have an antigen-presenting cell here. This antigen-presenting cell is going to run into a T helper cell at some point along its journey. This T helper cell, if it's the right T helper cell, will then bind to the dendritic cell in order to recognize the antigen that this dendritic cell is presenting. Here's more detail of the machinery involved. You'll recall that the T helper cell has this CD4 protein on its surface. The CD4 protein and the receptor, this cloverleaf structure here, carrying the antigen, will both bind to the MHC2 protein here. Once this has happened, and that antigen is recognizable by the receptor of the T helper cell, because remember this receptor was very specific, then the antigen-presenting cell is going to activate the T helper cell. The T helper cell will then release interleukins, which are a type of cytokine. These act as growth factors to stimulate the production of other T helper cells and many cytotoxic T cells. And this makes sense, because if we do indeed have an antigen that's recognizable by our immune system, then it's time to fire up the army of defenses, which include these cytotoxic T cells. In addition to stimulating release of interleukins to wake up all the other defense team members, these T cells will now go on to directly activate B cells. So we can then get B cells involved in assisting with the immune response. So this figure is a great review of how this presentation of antigens happen. Remember, so far we've had foreign molecules or microbes that have been flushed into, let's say, a lymph node. Here, an antigen-presenting cell, like a macrophage or dendritic cell, will pick it up and engulf it. It'll break down that microbe or other antigen into its component pieces and take the piece that's the epitope, the recognizable region, and mount it onto an MHC2 receptor. That MHC2 receptor will then be put out on the surface of that antigen-presenting cell. The cell is now presenting the antigen to a T helper cell. The dendritic cell will cycle through various regions of the immune system and eventually run into a T helper cell that's specific to the antigen that it's presenting. When that specific T helper cell recognizes this antigen, it binds to it. So now we have the antigen-presenting cell presenting its antigen to the T helper cell. The MHC protein and the T cell receptor bind together with the assistance of the CD4 receptor. This elicits the first stage in response, which is to have interleukins secreted, which activate other T helper cells, as well as cytotoxic T cells, so we can really elicit a response. In addition to that, it's going to activate the B cell system, which we'll take a look at here shortly. Before we move on to looking at how B cell response happens, let's take a look at an animation of this process.
Proteins generally require the cooperation of T helper cells, Th2, to stimulate B cells into becoming antibody-producing cells and memory cells. Such antigens are therefore said to be T-cell dependent. The protein antigen is first ingested and then broken into peptide fragments by an antigen-presenting cell, such as a macrophage. The antigen is then moved to the surface of the macrophage on a class II MHC. The T-cell receptor on the surface of a T helper cell recognizes the peptide antigen that is being presented on a class II MHC of the macrophage, and the T helper cell becomes activated. The activated Th2 cell is now capable of stimulating B cells. B cells also ingest antigens. The antigen reacts with an antibody on the surface of the B cell and is then internalized, digested, and presented at the surface of the B cell on a class II MHC. The antigen class II MHC complex of the B cell interacts with the T cell receptor on the activated T helper Th2 cell. The T helper cell produces cytokines, which stimulate the B cell to proliferate and differentiate into antibody-producing plasma cells and memory cells.
So now you can see how antigen presenting works, and also a little bit of a snippet of a view into the future, where we'll be covering how B cells are activated.